Hello, everyone. Welcome to the IWGSC Webinar series. Today, we're joined by Michael Scott, who's at the University College London, who will be giving us a webinar about NIAB populations. But before we get into our webinar, I'm going to give you a brief overview of the International Wheat Genome Sequencing Consortium, or the IWGSC. We are an international nonprofit. We have about 3,200 members representing 884 institutes in 71 countries. We have 7 sponsors that make it possible for us to do things like this webinar, and also that help us to lead efforts to advance genome resources for wheat. Our vision, IWGSC 2.0 vision, which is what we adopted after publishing the reference sequence in 2018, is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. In 2021, we are continuing a collaboration between IWGSC and Arbor Biosciences to develop tools and resources for the wheat community. We also plan to publish and release IWGSC RefSec version 2.1 and annotation version 2.1. Our goal this year is to secure funding and launch the IWGSC Wheat Diversity Project, which is to get platinum quality sequences for at least 8 land races that represent the breadth of diversity of the wheat gene pool. We also hope to be able to provide free publication releases of genome sequences for elite wheat varieties and other genomic resources that are developed by scientists around the world. And of course, we'll continue with our webinar series. It's unclear whether we will get to see anyone at workshops this year, but if not, we may have a virtual workshop at some point in the near future. Our next webinar, which we're doing in conjunction with the Phytobiomes Alliance, is titled Predictive Biology, Understanding and Reversing the Evolution of Antibiotic Resistance, and I would encourage you to join us with that. That will be held next week, and hope to see many of you there. So just a quick overview. The webinar is being recorded and will be posted on the IWGSC YouTube channel. And you can subscribe to the channel to never miss a new upload. The presentation section will be followed by a Q&A period. If you have any questions, please submit your questions in the Q&A panel. Don't use the chat unless you want to chat with someone else, chat with the organizers, etc. You can already download our presentations, both mine and Michael's, by going to the handout section. And with that, I'm going to turn it over to Michael Scott, who will give us a webinar on the NIAB Diverse MAGIC, Dissecting Trait Genetic Architecture Across 70 Years of Wheat Breeding. Michael, thank you very much for joining us, and I look forward to your webinar.
Thank you very much. So I'm just getting control now. Just give me a few seconds. You should see my desktop background. Okay. And now hopefully you're seeing the title slide. Okay, so I'm going to assume that's okay, and then begin. So I'm really pleased to speak in this excellent series, and so I want to say thank you for the opportunity to present our research to the organisers, and also thank you obviously to everyone for attending. I'm looking forward to hearing your questions or comments on this project. So I'm going to be talking about just going to show you my pointer as a laser. a multi-parent population of wheat developed at the National Institute for Agricultural Botany, NIAB, which is in Cambridge. And my role in this project was as a postdoc at University College London, and I was basically developing the genomic resources and analyses. So the overall project goal is to dissect trait genetic architectures in some diverse wheat varieties that span 70 years of breeding. So as I just mentioned, the goal is to look over a period of 20th century breeding and reveal the genomic basis of historical phenotypic changes, and then to assess the potential for future change, from that pool of genetic variation. So this goal means that you have to establish clear relationships between genotypes and phenotypes, and that's been called the holy grail of crop biodiversity studies. So to me, this term holy grail implies that it's a difficult task. So I'm just going to start by discussing the approach to linking genotype and phenotype in this project. So one way to confidently link genotypes and phenotypes is to establish an experimental mapping population by crossing. So a classic method would be to cross two varieties and create a heterozygote, and then create recombinant inbred lines from those through selfing. Each of these recombinant inbred lines inherits different parts of the genome from the two parents. So you can then measure associations between the genotypes carried and the phenotypes measured in these recombinant inbred lines, and that's what establishes the associations. In this presentation, I'm going to be talking about a multi-parent population, specifically a MAGIC population, which is an acronym for multi-parent advanced generation intercross. And in these populations, a number of founder varieties are intercrossed, often in a balanced funnel, like the one shown here, or in a series of funnels in our case. And this creates a highly heterozygous individual. And these are then selved to create recombinant inbred lines, which are the population where you're going to measure associations. So each of these recombinant inbred lines has mostly homozygous genomes that are mosaics of contributions from the founders. And there's two key properties of MAGIC populations. So first, there are recombination events captured across these multiple generations of intercrossing. And this means that these genomic segments tend to be smaller than they would be in a biparental population. And that therefore gives finer mapping resolution in these associations in MAGIC populations compared to biparental populations, all else being equal. And the second important feature is that there are multiple founders, obviously, in this population. So there should be more genetic diversity segregating. So I'm going to expand on that point a little bit now. So in these figures, the x-axis shows the minor allele frequency of a causal allele in a wider population of interest, which I've called the source population. And the y-axis is showing the probability of detecting this causal allele as a QTL in a mapping population of either 500 or 1,000 lines. And the detection probability is given by the probability that the allele is segregating at all, and the statistical power corresponding to the frequency it will be segregating at. So for these figures, I've just assumed that the founders are chosen randomly, such that a common variant only have a 50% chance of segregating in any particular biparental population. So here, that means you have to choose, founders with different alleles. However, if the causal allele does segregate in these, it has a high probability of being detected in a biparental population. So these are effective for QTL detection when the founders are known or suspected to differ for a particular allele of interest. So on the other hand, these MAGIC populations are constructed with more agnosticism about the target trait or alleles of interest. So this means you have a reasonable chance of capturing causal variation at any trait of interest. So if captured, none of the alleles are very rare, because the minimum they can be is 1 out of the founders at that frequency. So that actually increases the probability down here of detecting alleles that are rare in the source population overall. Nevertheless, this is still the most difficult type of causal variation to detect. And obviously you have a very good chance of detecting more common alleles as well. So MAGIC populations are useful, a generic tool for trait mapping, and they also, obviously, as they're experimentally produced populations, avoid confounding problems of population structure. So here we're using a MAGIC population to dissect sort of general patterns of trait genetic architecture in the population of interest. Okay, so this is the talk outline, and as we've just discussed, MAGIC populations have this carefully designed germplasm and a controlled population structure, and they're particularly powerful tools when that germplasm is combined with dense genotyping and phenotyping data. So I'm going to introduce the particular MAGIC population here by first describing the founders and the genotyping and phenotyping effort we performed on the founders and on the lines themselves. And later, I'll describe the results where we use a few different methods to link genotypes and phenotypes. Okay, so the diverse MAGIC population was constructed from 16 founders, which are in this table. And these were specifically chosen from a panel to capture as much UK wheat genetic diversity as possible. So that's why we call it the NIAB Diverse MAGIC. If you look in the second column, you can see that these founders were released in the UK over about 70 years. And as you can see in the third column, it's focused on UK diversity, but there are varieties from elsewhere in Northwest Europe. And the last column is showing the yield at the time of release. This is an interesting time span because the yields almost doubled, and a lot of that change has been attributed to genetic gain. So this is a diverse MAGIC experimental population that can be used to reveal the genomic basis of phenotypic changes that have occurred over this time period. Okay, so we sequenced all 16 founders after promoter and genic capture to 20x and called over 1 million SNPs. I compared these SNPs to a genotyping array data from around 4,500 traditional modern wheats from around the world, and found that almost 75% of the variants segregating on the genotyping array also segregate among these 16 founders. And that number is higher if you focus only on variants segregating in the UK or in Northwest Europe. So in summary, these are diverse representatives of the UK wheat gene pool. Now, one thing I noticed about this captured data and the SNPs from the founders is that at each locus, the founders could usually be categorized into 2 or 3 groups. So to quantify that, here I'm using a gene level analysis, so went gene by gene and included the promoters. And at each gene, I considered the founders to carry the same haplotype if they are all more than this percentage similar. So 99%, they have to carry 99% the same SNPs at that gene to be similar for this threshold, and then there's different thresholds used. And so what we see is that there's relatively few haplotypes. So using a 95% threshold, no more than 3 haplotypes at 90% of the genes, even though it's a diverse population that's capturing a significant fraction of global diversity as far as we can tell. So I'll also just mention that we used not just a gene level, but a larger block method and found a similar story. So I'm thinking that this basically indicates a low effective population size. So here I'm asking if that observation is a symptom of selection and bottlenecks that preceded 1935, which is the earliest founder in our population. Okay, so that gives you some information about the population founders, so the character of the population and the sort of variation of interest. And I've added this observation about there being relatively low haplotypic diversity as a result in this summary slide. And now I'm going to talk about the genotyping in the population itself. So this figure shows the pedigree, and these circles are the 16 founders I've just discussed, and they were crossed in a series of funnels. So 2 funnels are highlighted, one in pink and one in blue, and each combines all 16 founders in a balanced way, like I explained earlier. So after these crosses have been made, and then there's 7 generations of selfing, there's over 500 recombinant inbred lines, which we sequence at low coverage of 0.3x on average. And from that data, we're able to impute over 1 million SNP sites with high accuracy. So because I think possibly some of you might be interested in this imputation procedure, I'm going to describe it in a bit more detail. So we have 0.3x coverage of the inbred lines. This means that at any SNP in any line, there's less than 30% chance that we have any direct information about what the genotype is. So in this cartoon, I'm representing observed SNPs in white, and then the black is missing gaps. But we also have the full genotypes of the founders at over 1 million sites, and we're going to use those to fill in the gaps in the inbred lines. So from the observed genotypes, we can infer which founder contributed to each locus, found ancestry. So that looks something like this. And then we use the blocks of found ancestry to fill in what these unobserved missing genotypes are. So here the founders are basically serving a similar role to a haplotype reference panel. So this plot is showing what that looks like in practice in our population. So these are the 21 chromosomes of a single recombinant inbred lines, and the colors are showing the 16 founders. And the y-axis on these chromosomes is representing the inferred ancestry at each locus. So there's some loci where there's some uncertainty over which founder contributed that genomic region, but for the most part, the ancestry can be accurately reconstructed from this data. I find this quite pleasing because this is how we'd expect the chromosomes to look for some of these recombinant inbred lines. So for example, we infer about 5 recombinants per Morgan between founders, and that's roughly what we'd expect given those generations of crossing. And we confirm the accuracy of the imputed genotypes against, we use the genotyping array, the axiom wheat breeders array, and the concordance is over 99%. So we think the recombination mosaics and the genotypes can be accurately imputed from 0.3x whole genome sequence in this population. But we next did an in silico experiment to look at downsampling and see how low the coverage could have been, and also to do an experiment without including information from the founders. So in this plot, coverage has been downsampled to an average of 0.15x or 0.075x and so on. And we can see that even at 0.075x, which means less than 7.5% chance of observing any particular genotype, the accuracy of imputation is only slightly reduced. And the colors on this plot are showing 2 different imputation methods. So the first one in green is the imputation method I've described, where you simply project the founders onto the lines. The second doesn't use the founder haplotype information, so you don't use the founders as a haplotype reference panel. And here we're using Stitch software for imputation, and that is able to work in an iterative way where it infers recombination mosaics and then re-estimates the haplotypes that contribute to the population and so on. And so the orange is showing the accuracy from imputation using that method. So as you can see, even without final genotypes in orange, imputations at relatively high accuracy. So it could be an interesting genotyping strategy, even in populations without this particular structure or without an appropriate reference panel. Okay, so to say that again, we think that low coverage whole genome sequencing is a promising, cost-effective and accurate genotyping strategy in wheat, even without the reference panels available. The next key component that makes MAGIC populations powerful tools is the combination of the germplasm, the genotypes, and the phenotypes. So just briefly going to describe the phenotyping. So there's quite a big effort that I'm only briefly describing, and it was the phenotyping was led by Nick Fragiley at NIAB, who deserves a lot of credit for this effort. So there are over 500 lines, and they were in replicated yield trials with and without fungicide in 2 years. So that's over 2,000 lines per year. And there was 47 traits measured, of which 25 were measured in both years. So 72 phenotypic measurements that we're going to look at. I won't go through all of the traits or measurements, but hopefully this table gives you some idea. I've just roughly categorized them into sort of yield-related, yellow rust resistance, some phenology-related traits, seed, growth habit, and so on. Okay, so now we have these important ingredients. We have over 1 million SNPs, over 70 phenotypes, and over 500 lines with controlled structure. And we want to use them to assess some general patterns of trait genetic architecture across around 70 years of breeding in Northwest Europe. So the first common method to do that is QTL mapping, which you're probably familiar with. This is a Manhattan plot for a single trait, which is sort of representative, which is plant height to the ear tip in the second year. The x-axis is showing the position of SNPs along the 21 wheat chromosomes. And the y-axis is a measure of the strength of association between those genotypes and the phenotype. And in this case, there are some loci which have a significant association with height, according to these genome-wide significance thresholds. So now I'm summarizing the results of that. That was just for one trait. I'm summarizing this analysis over all the 72 measurements, which are just roughly categorized again and coded along the bottom. So the lollipop is showing the trait heritability based on genotypic relationship between lines. And the bars are signifying the fraction of phenotypic variance that can be explained by all the QTLs that have reached genome-wide significance. So in the example I showed before, it'd be those ones that go above the thresholds. How much phenotypic variation can those explain combined? So across different measurements, we can see a range of genetic architectures. So I'm highlighting here, so for some traits, almost all the heritable variation is accounted for by a small number of genome-wide significant loci. So for example, the presence of orns, some pigmentation measurements, vernalization requirement, that's spring habit, and this is pre-harvest sprouting score, are traits like that. And there are some other traits where there are QTLs of a large effect, but also a reasonable amount of heritable variation that's not accounted for by the genome-wide significant QTL. So that's the case for example, height and some flowering time measurements. And finally, I'm pointing at some traits where there's either no loci that reach genome-wide significance. This is green leaf area measured at different time points, or the QTLs only account for a small fraction of the heritable genetic variation. And these is grain yield and protein content here. So I think it's nice that we have all these different phenotypes with different genetic architectures, varying from a Mendelian trait to polygenic traits, and they're all in a single well-characterized population where we can compare them. So that's one thing. I'm going to summarize this QTL analysis in a different way now. So this is a different summary of the QTL. So there are 136 significant associations between genotype and phenotype, but these are actually grouped at only 42 unique locations. So quite a lot of the QTLs are affecting multiple of the phenotypic measurements. In this plot, the x-axis is the peak p-value of association with the phenotype, and the y-axis is the mapping interval size, so the plausible location of causal variance that we've set. So that's to give you some idea of the mapping resolution, which is generally smaller for larger effect loci. Yeah, it ranges, as you can see here. It's a log scale, but reasonable in some cases. Each point is a locus, one of these 42 loci, and the size of the point indicates the number of phenotypes where we get a significant hit. So there's a fair amount of pleiotropy between different phenotypic measurements that we've taken. Okay, and the colors in the plot show our analysis of co-localizing these genes with previously reported QTLs. So we've just categorized them into high and low confidence, depending on the nature of the evidence for overlap with previous studies. So for example, a low confidence co-localization might be, have found a QTL in the area of a genetic map, which roughly corresponds to the area of the physical map that we've mapped something to. And then, and in all the cases where there's a strong candidate gene known, previously reported, it falls within our mapping interval. So that's some validation of the calibration of these interval sizes. So you may have noticed that we've captured some of the most well-known variants, such as green revolution alleles or photoperiod genes for latitude firing time. But generally, I think the fact that a substantial fraction of these QTLs can be co-localized in this diverse population is partly a function of the fact that this is a relatively well-studied gene pool, but perhaps it could also reflect the overall low gene level haplotypic diversity among the founders that I mentioned earlier. Okay, so the QTL mapping there has shown a variety of different genetic architectures for different traits, varying from Mendelian to polygenic. And next, I'm going to discuss genomic prediction for those same traits in this population. So genomic prediction is a method for looking at some of the genetic effects outside of those genome-wide significant QTLs identified in the previous analysis. And I want to mention that these genomic prediction models were fitted by my lab mate, Funmi Ladajobi. So the top panel is the Manhattan plot for plant height that we saw before. And the bottom panel shows a subset of around 100 SNPs that were chosen by a lasso genomic prediction model to predict plant height. So the y-axis is showing their estimated effects on plant height. So the model is including some of these large effect loci and also a range of smaller effect loci across the rest of the genome to predict plant height. So we evaluate the accuracy of these prediction models by fitting the model in one portion of the inbred lines and then predicting the phenotype of the rest of the inbred lines based on their genotypes only, and then assessing the accuracy in that set. So this plot is showing the prediction accuracy. Each of the points is a phenotype, and the x-axis is showing the fraction of phenotypic variation that was explained by genome-wide significant loci. So the prediction accuracy generally gets higher when for traits where there are large effect QTLs around. And overall, prediction accuracy is varying, but it's on the whole reasonably accurate. So the correlation between prediction and real phenotype is 0.43 on average across the different traits. This next panel is showing the number of SNPs in the lasso prediction model against the estimated fraction of heritable variation that can't be accounted for by a small number of large effect QTLs. So it's kind of unexplained heritable variation. And a higher number of SNPs doesn't necessarily mean higher accuracy. What this is reflecting is the genetic architecture of different traits. So the model is including a larger number of SNPs when there's a larger fraction of, it's using a larger fraction of the genome to predict some of the more polygenic traits, essentially, where there's lots of heritable variation that can't be accounted for by QTLs. Nevertheless, the lasso model is always selecting a relatively small number of SNPs and giving a reasonable prediction accuracy for the trait, which I thought was quite interesting. Okay. Next, we took the genomic prediction models and used them to assess the potential for further improvement, using the genetic variation that's segregating. So in this plot, the green densities are the predicted breeding values for the real test set of lines here. So these predicted breeding values show some shrinkage back towards the mean, as expected for these type of models. In orange, I'm showing a simulated population that's much larger. So it's 20,000. It's simulating the creation of 20,000 recombinant inbred lines. And the predicted breeding values for these imaginary simulated lines is basically overlapping with the distribution for the real lines. However, the extremes, which are shown by these vertical lines, are slightly more extreme. And that's because you're creating new allelic combinations. Among this 20,000, there might be one that happens to have lots of positive effect or negative effect alleles. And then the purple line is showing the extreme that would be possible, based on the models. So that means if you were to combine all the positive effect alleles or all the negative effect alleles, what does the genomic prediction model predict about the most extreme phenotype possible? Okay, and then this right-hand panel is basically using a similar principle. So these are the three. This is plotting the three phenotypic extremes. So essentially the green line, the orange line, the purple line, for all the different traits. What you can see is that the extreme of this much larger population is only a little bit more extreme than the extreme of the real population. So basically, so it's 0.5 standard deviations more extreme on average. So that reflects the fact that it's difficult to randomly create a combination of positive or negative effect alleles. Whereas the model is predicting large phenotypic shifts are theoretically possible, but these will require the selection of dozens of loci. So overall, I think these genomic prediction results are consistent with the idea that genetic gain is difficult to achieve, and it's taken many years, from this gene pool to fix various and reshuffle various positive effect alleles. Okay, so I've added these results that we have the prediction accuracy, rough idea of the number of SNPs involved in the lasso prediction models. And the last thing I'll describe has to do with the relationship between different phenotypes. So this plot is showing the yield in each year against the protein content. And the founders of the population are the larger dots that are colored by their release year. So you can see that the more recent varieties, the more yellow ones, tend to have higher yield. However, they also tend to have lower protein content. And there are a few also, some even more recent than the population founders, which are shown in yellow. So for comparison, and they are essentially continuing this trend. And then the black dots are the inbred lines. So despite the fact that the genomes of those founders have been shuffled, the trade-off between yield and protein continues in the lines, suggesting it might be difficult to avoid. However, we wanted to look at what the genetic potential was for breaking this trade-off. So we looked at a composite phenotype, which is the deviation, protein yield deviation. So that means the perpendicular deviation from a symmetrical regression between yield and protein. So that's what we're looking at now. These histograms are showing the values of deviations away from the trends between protein and yield. And the colors are showing that the lines with and without orns, where the orns lines tend to have a higher mean, higher than expected protein than you would predict from their yield, or equivalently more yield than you would expect from their protein level. So the orns locus, which is a Mendelian trait, is here on chromosome 5A, and it reaches genome-wide significance if we combine both years to make our association. So if replicated, this might be an interesting locus and phenotype to target towards breaking this trade-off. I'm also showing the heritability and prediction accuracies obtained for this phenotype, the protein yield deviation, which indicates some modest potential for genetic variation segregating that you could use to break this trade-off if you were to target it in this way. Okay, so that's everything I specifically wanted to say, and now I want to look across all the things I've shown and try and draw some conclusions for us to think about. First, we saw that low coverage whole genome sequencing and imputation could be a cost-effective and accurate genotyping strategy with coverages much less than 1x, and that's even without the use of even where haplotype reference panels aren't available. So I'd be interested to know if this strategy will also translate to other samples or populations. Second theme relates to the value of the multi-phenotypic approach. So this allowed us to identify traits with different genetic architectures, and then also to compare genomic prediction accuracy or different genomic prediction models on different traits. And this is within a controlled population where we can make some kind of comparison between traits. And finally, obviously, the last story I told you about the trade-off between yield and protein and the potential association with Orn's locus relies on the fact that you have measured multiple traits in the same lines. So I just highlighted the value of the genotyping approach and the phenotyping approach. And finally, I'm highlighting some of the results that I think indicate how challenging it can be to make genetic gains using this segregating genetic variation. So given the largely polygenic nature of yield, the breeders have obviously done a good job of achieving genetic gain, but it appears, based on these results, to have involved the ever finer reshuffling of a limited number of haplotypes to combine those positive effect alleles. And of the loci with large effect, many have been identified, which perhaps indicates that there aren't a lot of easy wins left to be fixed from within this pool, as far as we can tell here. So these results make us ask the question whether we should use a strategy where we are selecting from within this pool of genetic variation, while also targeting particular alleles or lines to introgress from more exotic or different germplasm. Or would the best long-term strategy be to obviously expand the segregation haplotypic diversity genome-wide, or would that just simply prove counterproductive? So that's obviously a bit of a big and difficult question, and possibly also a bit above my pay grade and speciality. But I wanted to include that as some food for thought in light of the results that we have. Okay, so now I want to acknowledge some people. So the construction of the population was started over 10 years ago at NIAB. And so these are some of the people that were involved in the conception and design and supporting different phases of the population. And I've highlighted Nick Fragile, who I mentioned, who's also the first author for this preprint and led the phenotyping effort and went the extra mile with that to create a range of interesting phenotypes. This is my postdoc supervisor and lab mate at UCL, Fiona Cork at Aberystwyth, provided some extra phenotyping data, while Thomas Brabs at Earlham performed the captures for the founders. Okay, and this population I've described has this combination of germplasm, dense genotyping data, and dense phenotyping data, plus associated software tools. And the aim of all that is to create a useful, a powerful community resource. So, for example, as I mentioned at the beginning, it's reasonably likely that a trait that you're interested in might have some genetic variation segregating in its population. So you can obtain the germplasm, measure that trait, and then we provide the data and some functions that you could use to do mapping on that trait. I think there's lots of other interesting things that you could do with this data. We've done an initial analysis that I've described today, which you can look at in more detail in this preprint, or for more information on research and applications of multi-parent populations in crops, you could look at our recent review as well. Okay, so with that, I'm going to finish and take questions, and I'm happy to obviously answer any of your questions. Thank you.
Great, thank you very much, Michael, for a really interesting webinar. We have a lot of questions have come in, and I'm not sure that we will get to all of them, but okay, rest assured they will be answered, and we typically try to post them along with the video of the webinar. So if you don't get your answers today, then you will either hear directly from Michael or your answer will be listed in among printed questions. So the first one goes back to the beginning of your presentation. How were the haplotypes called at the gene region?
Yeah, so that's just a simple analysis. So you take all the SNPs in a gene and promoter, and then you say, okay, for something to have the same haplotype, it has to be more than 90% similar with all the other ones that carry that haplotype. And so those different thresholds I showed were just different thresholds for how similar you have to be in the same haplotype. So that means, if there's 100 SNPs, that at least 90 of them are the same between having those lines that have the same haplotype. And I varied the threshold obviously in that figure.
So
that's clear.
Yeah. How did you do the QTLs? How did you identify the QTLs in the multiple year studies?
So we didn't combine. I did the association on the different years separately. I think that's probably what the question there is asking.
Yeah.
I don't know if I want more detail on the process, but I will just leave it at that, I think. Yeah.
Okay. Let's see. When you select the founders, do you have some traits already in mind, or do you just take random cultivars regardless of the specific traits or genes for which they are known?
Yeah, so the founder selection was done before my involvement, but basically there's a panel of about 100, I can't remember exactly, maybe 200 UK varieties, or varieties released in the UK, that had at that time, you know, a few hundred genetic variants in them, and they used basically a stepwise algorithm for selecting as much diversity from within that panel as possible. So it's not actually targeted towards you know, the characteristics of those beyond getting as much diversity as possible.
Right. Well, that seems to make sense. I mean, if your goal is to enhance the diversity of the germplasm. Yeah. So what is the minimum value of genomic prediction that you would consider to be good?
Yeah, it's a good question, and I'd actually also be interested in knowing the answer, because you know, I'm obviously not involved in breeding, so I guess the answer to that question depends on how much gain you're getting from maybe making your selections based on genotype and therefore potentially having more lines, or making your selections quicker or earlier, versus the loss of accuracy you get from using genomic prediction. So, I mean, it's not a, you know, I can't personally put a figure on that answer, but yeah, maybe I have to ask somebody else.
Okay.
Yeah.
Can you explain the heritability in R2 plot a little bit more?
Yeah, okay, so I'm going to bring it back up just because there's a specific question about that. Okay, so I think you can see that now. So the heritability is estimated from the, you know, genetic relatedness, the kinship between lines. So that's a genome-wide measure of the fraction of, this is phenotypic variance that is estimated to be accounted for by all of the genetic, you know, all of the related, the kinship between all the lines. And the bar means, if I just flick back to this one, I take all of the loci that exceed the genome-wide significant threshold, I fit those against the phenotype together, and it's the joint amount of phenotypic variation that those, you know, say for example, these three loci can explain. So that would be height to ear tip in year 2. They can explain about 50% of the phenotypic variance, whereas the genome-wide estimate of heritability is closer to, I don't know, looks like 90% or something. So hopefully that's a bit clearer.
Okay. What tool did you use and would you want to use for variant detection?
Okay, so we did the variant detection and filtering among the founders. So in that case, we had exome and promoter capture data, and we had 150 base pair paired end reads from that. And in most cases, I found that was sufficient to distinguish between homologs in terms of the mapping quality. You know, and then I basically just did the kind of relatively standard filters and calling procedure I implemented in GATK software. Yeah, and then another thing I did was so you're worried about variants from other homologs giving you false sort of SNPs. And the idea is that a lot of the time that is going to give you maybe a heterozygous call. It looks like a heterozygous, but really you're just missing some variant and it's mapping from a different homolog or something. So I removed all heterozygous calls and did some other aggressive filters. Yeah, so there's also no missing calls in the founders. So those 1.1 million SNPs are I didn't look at any indels, so they're kind of quite aggressively trimmed, just basically tagging markers for this purposes.
Okay, so was your goal to find markers linked to complex traits or to develop lines for breeding?
Well, I think that you know, these type of mapping populations, a core goal is to find associations between genotypes and phenotypes that could be maybe used for markers. But I do think it would be interesting to use multi-parent population germplasm for breeding. I'm not sure if this particular population is well, you know, you'd have to make a few years of selection in order to catch up to the more modern varieties. But that's something we did discuss in a few other crops in this review. In some cases, you know, MAGIC population germplasm is being you know, fed into breeding programs as pre-breeding material. You know, this germplasm is freely accessible to breeders, so they can select lines and use that in their program. And in some cases, like in chickpea and rice, and possibly some others that we highlight in this review, people have taken MAGIC lines that combine, say, multiple resistance loci because they've been reshuffled, and then release those as varieties in themselves. So yeah, it's an interesting thing to do.
So would future phenotyping necessitate using all 500 RILs, or could you use a subset of 150 lines?
Yeah, it would be, yeah, because obviously some phenotypes are challenging to measure or other constraints. So the only problem if you were to use less lines would be that you're reducing your power and potentially mapping resolution. So it's a trade-off between the practicalities of measurement and the power that you have. So, you know, obviously with lots of studies, it's a good idea to do a power analysis in advance yeah, for that.
You may not know the answer to this, but have any of these lines been grown outside of the UK?
I guess that means the inbred lines.
Yes.
Yes, I think yeah, that would be something that is so no, this is everything I've described is from the NIAB site, is phenotypes measured at basically a single site. But again, that is something that is interesting in multi-parent populations more broadly. If you look at multi-environment associations or, you know, trade-offs and things, so there is that's also discussed in this review. As I remember, there's a tomato population that's focused on doing that, and some of the rice populations are specifically focused on, say, heat adaptation, so that, you know, you're obviously wanting to measure those same lines in multiple environments and then do your associations on their sort of response to different environments.
All right, you mentioned you tested your genomic prediction on an independent set of lines. Can you give more information about the lines? Have they been, well, this kind of continues, have they been evaluated on the same environments as the MAGIC training lines or different ones?
Yeah, so yeah, so actually the way that the genomic prediction accuracy was evaluated was kind of the best case scenario. So all we did is cross-validation in the same data set. So we did 50 replicates of 10-fold cross-validation, I think. So there you're just splitting the same lines into different sets of training and test set. And so you're predicting kind of somewhat into the same environment, and you know, you're not predicting outside of the MAGIC population, for example, into a set of breeding lines. So obviously with a genomic prediction, the more distantly related the test set is from the training set, that you'd expect the prediction accuracy to go down. So you would definitely want to be cautious or skeptical about just taking this prediction model and just applying it to a different population completely.
All right. Can you explain more how you guess the missing sequences of the progenies without those of the founders?
Okay, so I'll just return to that slide again. So probably just went a little quick over this. So, okay, so imagine that we don't have these to begin with. What this software does is kind of it just starts from somewhere at random, and then you're predicting these mosaics and filling in the missing genotypes, and then kind of assessing how accurate that was. And then you use, any discrepancies that you've seen, like say, for example, these observations might not be possible given the set of founder haplotypes. So then you would go back to this haplotype panel and update that. And then now you've got an updated version of the haplotypes. You go back and you estimate the recombination mosaics now, and you're constantly assessing how many discrepancies you have between your data and your sort of full genome mosaics. So basically, by iterating that over a number of cycles, you converge on a sort of a set of haplotypes that can be combined to explain the data that you have through various recombination events.
All right, can you?
Yeah, you can see that this is the paper to look for that information. Sorry.
Can you explain how you make the differences between the high confidence and low confidence co-localization QTLs with known genes?
Yeah, so basically it's a manual process of trying to look for candidate, previously reported candidates that are at a similar position to the ones we've found. Okay, so we could have missed some or something. And then you say, okay, so this previous study, what kind of evidence do they have? And in many cases you have, let's say you have a strong candidate gene on the physical map. Then we can say, okay, is that within our mapping interval or not, affecting the same traits? And in other cases, they might have preceded the physical map. The QTL study might have preceded the physical map being available at all. So then what you have is genetic map location, which is a QTL, say, at a genetic map location, which is near the beginning of a particular chromosome. And so there you think, okay, this is probably the same QTL that we've identified, but you can't assess, is that locus within our mapping interval? Because it's not comparable. So that's an example of what we call high and low confidence co-localizations.
Okay, one last question. As you look into the future, and particularly based on the review that you did of multi-parent populations and other crops, what do you see as the future or the most pressing need for wheat?
Okay, so I'm not sure if this is a question related to, well, I guess it's related to multi-parent populations. So
well, not necessarily, but it's with regards to breeding generally. How do we make investments, and how do we actually really break this protein yield problem?
Yeah, so hopefully, some of the results from this are interesting in that, I've discussed, but I'll just mention one thing this makes me think of, which is that, we designed this at diverse germplasm, and then we had this interesting thing of at the very fine level, maybe there's not that much haplotypic diversity. So, and I suggested maybe breeders are sort of reshuffling a small number of haplotypes to create these positive effects. So if you're thinking in terms of magic populations, it might be interesting to try and expand that a bit. Now, the problem with that is, are you just going to then map lots of traits which are relating to the adaptation to the very dispersed, disparate environments in which you've selected the founders from or something? So there are a few ideas for how you might do that, like there's something called a magic mirror, for example. But so yeah, I think that would be interesting, an interesting way to go. And as I mentioned, when I looked at these results, I thought, maybe we should try and up this haplotypic diversity more generally. I'm aware that might introduce some things that you don't want, but that's what it made me think, definitely.
All right. Well, thank you very much, Michael. Again, just to remind everyone, the questions you'll be provided with answers if we didn't get to your question today. And we look forward to seeing you on our next webinar. And again, thank you very much, Michael, for a great presentation. And we look forward to your future work.
Thank you very much again.